Thank you so much to all the organizers for inviting me to share the work of my lab. And of course, to all the people joining into the seminar series, especially because of the holiday, as Sonia pointed out. So my lab is interested in understanding how organisms harness spatially organized mRNAs to promote development. And so these RNAs, they're also called localized RNAs, are important because they allow the formation of protein concentration asymmetries within the developing organisms. As the RNA localizes, translates somewhere, it builds up protein, and this really helps to create asymmetries in protein concentrations that then lead to asymmetries in function as well as in shape of the developing organism. And this is observed across organisms, especially in the early embryogenesis. This mechanism is quite prevalent. And here I'm showing an example of early Drosophila embryos. These are different embryos collected, where in green you can see different mRNAs stained by fluorescent C2 hybridization, which reveals a localization of these RNAs through the embryo. I hope you can see my pointer. I'm going to be using it through the talk. And then in magenta is our nuclei, the nucleic acid stained by DAPI. And so in these embryos, more than 30% of maternally transcribed mRNAs are deposited into these embryos, and they're localized somewhere and in some pattern. And these patterns are important because they are driving early developmental decisions, including patterning of the embryo and the formation of first cellular lineages. And many times, mRNAs are going to organize and localize within biomolecular condensates or RNA granules. And here I'm showing you some of the examples, such as stress granules, P-bodies, germ granules that we work on, nuage, and I'm pretty sure that these are condensates that are known to this audience. And so what is known about these condensates is that they actually share core features, the way they're forming through condensation. And they are full of RNA binding proteins and mRNAs, and they are thought to be involved in very important post-transcriptional regulation processes. For example, stress granules are thought to regulate and protect mRNAs during stress. Processing bodies or P-bodies are thought to be the sites of mRNA decay in cells. Nuage, shown here at the bottom, is thought to be the site of piRNA biogenesis. Germ granules are accumulating mRNAs that are involved in the specification of the germ cell fate. And so in my lab, we're trying to understand what these granules actually do, how they're regulating their mRNAs, but also how they form. And knowledge through the past 2 decades has really shown that these condensates are relying on the multivalency of interactions. And this multivalency involves both proteins and mRNAs. And they, of course, are involving protein-protein interactions, protein-RNA interactions, as well as RNA-RNA interactions. And so we're trying to understand also in my lab how these interactions are driving condensation. And today, and for majority of my time since I started my lab, we've been focusing our attention on the involvement of RNA-RNA interactions, and specifically intermolecular base pairing. So what is known about intermolecular base pairing in vivo? Actually, not so much. We don't really understand the prevalence or abundance of these interactions, but what we do know is that these type of interactions are quite common in RNA viruses, such as retroviruses. For example, HIV virus uses intermolecular base pairing to form an RNA dimer, and this RNA dimer is basically its genome that then the virus packages into a virion when it's budding from an infected cell. And so the way this base pairing occurs is that the virus itself has a small stem loop structure at the beginning of the RNA, and at the top of this stem loop structure is a GC-rich palindrome. And this palindrome engages in kissing interactions involving base pairing interactions, and this is really what drives dimerization. And so, as I said, this is prevalent in RNA viruses, but we don't really know much about it in eukaryotes. However, there are a few examples. In Drosophila, an RNA called OSCAR uses this type of interaction to become localized at the posterior tip of the developing oocytes, and I'll tell you a little bit more about it later on. And more recently, particularly Parker and Alberti labs showed that intermolecular base pairing plays important roles in the formation and condensation of RNA granules, stress granules in particular. However, when these interactions become too many, exaggerated, they can also lead to disease, and this is really associated with trinucleotide repeat expanded mRNAs. So here I'm showing an example of a myotonic protein kinase, a myotonic dystrophy protein kinase gene that has CTG repeats in its 3' UTR. And when these repeats become enhanced, so repeated between 100 to 4,000 times, so these are mutations that occur naturally, then these mRNAs are transcribed, and instead of being exported into the cytosol where they're translated, they engage in interactions with neighboring mRNAs that have they share the same type of expansions, and they form nuclear foci. And these nuclear foci, they're called toxic nuclear foci because they're generating disease. So this is in case of myotonic dystrophy 1 disease. This is an RNA gain function mutation where these foci really are the causative agent of disease because they're sequestering a variety of splicing factors, which then messes up the normal biology of the cell. So this is really causing then progressive muscle weakening as well as premature death. So the recent simulations of how RNAs in these repeat expanded diseases are forming RNA clusters have revealed that these RNAs that contain repeats are normally folded when they're monomeric. However, when they're assembling and forming clusters, they unwind. And this unwinding then allows them to start interacting intrans, so intermolecularly, using base pairing. And this enhances, of course, clustering. However, it also, when in the cytoplasm and exposed to the ribosomes, can interfere with normal translation. And this is also part of the mechanism by which this intermolecular base pairing, when it's enhanced, can interfere with normal biology. So we, in the lab, are trying to understand the prevalence of these interactions and how they are being regulated and how they occur. And today I'll try to tell you two stories. In the involvement of this intermolecular base pairing, first in the formation of mRNA transport particles. So these are RNAs that are becoming localized. And I'll tell you a little bit about this, but then a little bit more about RNAs that are found in germ granules. So these are localized RNAs. They have reached their final destinations. And I'll tell you our findings about the involvement of base pairing in these localized RNAs as well. And so to understand how base pairing among mRNAs is regulated and how it occurs in vivo, we turned our attention to OSCAR RNA. I showed you previously that this is an RNA that localizes in Drosophila. And here I'm showing an outline of a Drosophila embryo. It has an anterior and posterior end. And here below, I'm showing you an image of this embryo that was stained with OSCAR mRNA fish probes. So we are using fluorescence in situ hybridization in the lab to characterize these mRNAs and how they localize and how they cluster. Specifically, we're using DNA probes. These are short 20 nucleotides DNA oligos that are covalently bound with a fluorophore. So these are commercially available DNA oligos that are not that expensive. But the utilization of many of them, so in this case, we used 48 of them, which are hybridizing to different parts of the RNA, really amplifies signal to noise ratio and allows detection of the RNA. And so this is what we see with this RNA at the posterior. So you can see that it's localizing and enriching at the posterior. And as I said before, the way this RNA localizes is in part by using a molecular motor, which brings the RNA to the posterior. But then the RNA uses this intermolecular base pairing driven by this palindromic sequence that is found at the top of a stem loop structure to enhance its localization, because the RNA piggybacks on the first. So this is similar to what I showed you previously for an HIV RNA. So, but what is interesting, so this is how the RNA reaches to the posterior. However, if you zoom in with your microscopes to the region of the embryo that does not contain localized RNA, so these are so-called somatic regions of the embryo, you can see the following pattern of Oscar RNA. It has clusters that have more or less fluorescence intensity, which indicates that these are clusters that have more or less Oscar RNA. So this RNA is already organizing as a transport particle before it reaches the posterior. And you can see within the heat map image here how this abundance of this RNA is distributed among these RNA clusters. So you can see singlets, doublets, clusters that contain 3 or more mRNAs, and even some clusters that contain more than 11 Oscar RNAs. And so what we have found recently is that the way these clusters form is they make dimers, and then they make oligomers that contain more than 2 Oscar RNAs. But in order to make this process happen, 3 things need to cooperate with each other, or basically they're working with each other. The process relies on high RNA concentration. It relies on this the palindromic sequence, and of course RNA binding proteins that kind of glue everything together. So this augments dimerization. However, the formation of an oligomer actually relies on RNA binding proteins that are multivalent. So Stauffen RNA binding protein has 5 RNA binding domains, and it seems that it's able to grab different RNAs together, enhancing oligomerization. So the palindrome enables dimerization because it's monovalent, and then multivalent RNA binding among them is Stauffen are then helping to drive oligomerization. But there is another interesting thing that we observed, and that was that the palindrome is not only important to drive the initial dimerization of Oscar RNA, but it's very important for determining compositional specificity of the dimer and ultimately of the oligomer. The way we observed that was that we generated flies that co-expressed along with the endogenous Oscar RNA a transgenic RNA that carried an open reading frame of the GFP sequence, and it actually contained this palindromic sequence, the stem loop structure of the endogenous Oscar. And we observed, we were able to label both RNAs with green and magenta, that they were colocalizing with each other, so they were co-organizing. But when we mutated the first 2 cytosine for adenosine, in this palindrome, this was sufficient to really reduce colocalization between the transgenic RNA and the endogenous RNA, really indicating that this base pairing is not only important for formation of dimers and oligomers of this RNA, but it's also very important for driving compositional specificity of these clusters. Okay. So what about RNAs that have reached their final destination? So these are localized RNAs. How are they interacting with each other using base pairing interactions? So to do this, we looked at germ granules, and the way these granules form is, they form at the posterior tip of the fertilized embryo. So I showed you before that Oscar RNA localizes to the posterior. It then translates there. And this translation of this RNA, which generates Oscar protein, ultimately drives the formation of germ granules, shown in green. And these germ granules then recruit mRNAs. And so these mRNAs also become translationally active in these granules. And this is important because the protein products from these mRNAs are then driving the formation of germ cells and the segmentation of the body plan of the embryo itself. And if this process is somehow perturbed, then then these germ cells do not form and the embryos does not segment, which really leads to infertility and death of the offspring. So it really allows us to study these processes with a really nice functional significance. And that's great. But the reason we also really like this system is because within these granules, when RNA is localized, they also form clusters. And this is an example of nanos RNA. So this is a heat map image of an SM fish stain nanos RNA. And you can see that outside of granules, this RNA is mostly present as a single transcript. However, within clusters, within these granules, it actually forms clusters. And these clusters have various abundances, ranging from a singlet to more than 11 mRNAs. The concentration of nanos RNA within this clusters is also very, very high, more than 5,000-fold increase relative to the single RNA outside of granules. So this really indicates that these RNAs come into close proximity to each other, which may augment interactions among the RNAs. And nanos is not the only RNA that can cluster. Other RNAs that localize to these granules also can cluster, cyclin B, PGC, and GCL, for example. But interestingly, this clustering behavior in germ granules has been observed in other organisms, such as zebrafish and C. elegans. So it's a conserved behavior of RNA within these granules, but also in other RNA granules per se, including P bodies and C. elegans. And we've also recently observed them in stress granules in human cells. So this clustering behavior is just something that RNAs do under certain conditions, and those certain conditions probably are related to just high concentration. And we're trying to understand what is the significance of this clustering and how is clustering generated. However, and this is very interesting, but it was also a little bit alarming because I showed you before that RNA clustering typically is associated with neurodegeneration. So with these repeat RNAs, which are forming toxic RNA foci within the nuclei of affected individuals, these are really RNA gain-of-function mutations. And yet, we observed clustering of RNAs in the very granules that are required to generate the next generation of an organism. So clearly, it's suggesting that if interactions occur among these RNAs, that their abundance likely is somehow regulated. And this is what my student Saran Tien was set out to test. And initially, the way she approached this problem was to generate two mRNAs, two transgenic mRNAs that had two different open reading frames, so we could detect them differently with two different sets of SM fish probes, but they shared 500 nucleotides of perfect sequence complementarity. So what we were expecting was that the two RNAs are going to come to the same granule and colocalize because they're going to base pair. But that's not what she observed. So she observed that the two RNAs came to the same granule, but actually very poorly colocalized. And for comparison, an RNA that's labeled with two sets of probes has a very high colocalization. So clearly, these two RNAs were not interacting with each other, at least not very efficiently. And for another comparison, I showed you before that a single 6 nucleotide long exposed palindrome is sufficient to co-organize endogenous Oscar with a transgenic RNA, whereas this 500 nucleotides is not sufficient within the context of germ granules. So it really was suggesting that these interactions, should they occur, are probably regulated. And so the way they're regulated at the time was not known, but we were speculating that perhaps they're unavailable for interactions, the sequences, and maybe that is done through RNA structure. And that's what Saran was set out to test. So we collaborated with Sylvie Ruskin's lab at Harvard, and we used their DMS-MAP-seq approach. So DMS is a reagent that, when in contact with an RNA sample, will form little modifications on exposed and unpaired A's and C's. And those modifications can then be read through reverse transcription to generate a reactivity profile with which you can then predict the secondary RNA structure of an RNA. And so we homogenized a lot of embryos and then purified granules through several rounds of centrifugation. And we obtained an RNA fraction that was localized to the granules, but we also obtained an RNA fraction that was not localized. So about 4% of an RNA localizes to the granules, the rest remains unlocalized. So we were able.
Tatjana, just to let you know, about 4 or 5 minutes.
That's great. Yeah. So we were able to compare the two structures before and after localization. And we specifically looked at the 3' UTRs of these RNAs because the RNAs are translating, so the ribosomes would actually just mess up the reading of these modifications and the structure itself. So here I'm showing you the 3' UTR of unlocalized nanos RNA. You can see that's very structured. And here in the box, I'm highlighting the translational control element that I'm enlarging here. And this is important because the same structure was before predicted computationally by Liz Gavis's lab, so we were able to recapitulate it, but also because the structure binds a translational regulator called SMOG. So we can probe this structure even within a proteinaceous environment. We then looked at the reactivity of the RNA inside and outside of granules. So we calculated the fold change, and you can see that there's not much change in the reactivity. So the RNAs are not massively unwinding or changing upon their localization. In fact, this is how the RNA looks like within the granules when it's clustered. So it's not a lot different. So this model of clustering is very different from repeat RNAs, which when they cluster, they're unfolding. We're finding that RNAs that are localizing to germ granules and Drosophila remain structured and remain folded. So this was great, and we were excited about this result, but it was a little bit at odds with some of the in vitro experiments that other labs have observed, including ourselves, where RNAs can interact with each other in vitro, even in the absence of proteins. So maybe they're utilizing type of interactions or base pairing interactions that cannot be experimentally accessed, but maybe we can predict them computationally. And for this, we collaborated with Dave Thirumalai's lab at UT Austin, and at a time his postdoc, Hang Nguyen, who now runs his own lab at the University of Buffalo. And so they use simulations to predict how these RNAs are folding when monomeric and dimeric. And here I'm showing you a 3' UTR of nanos RNA. You can see that's very well structured. But surprisingly, this remains true when the two RNA form a dimer. So one nanos 3' UTR and the other. So they're interacting, and yet they're still very, very structured. In fact, the majority of interactions, base pairing interactions, occur within the molecule itself, so they are intramolecular, and only a small percent occur between the RNAs. And these interactions occur with low base pairing probability, and they use sequences that have low sequence complementarity. And so what we were able to determine using all of these experiments is that and strictly looking from intermolecular base pairing point of view, we're disregarding proteins at this moment, that RNA clusters form through base pairing interactions that occur on the surface of RNA. So these are surface exposed nucleotides. They are infrequent, they are transient, and combination of this nevertheless provides multivalency as well as malleability of the cluster. And if I can compare the two systems now, the RNA transport particles that are carrying the RNAs to the destination, so these interactions are persistent, they occur with low multivalency, but they're also not very malleable. Whereas in granules, these clusters that are forming where the RNAs are translating and have already arrived to their destination, these interactions are transient, they are highly multivalent, but they're also very malleable. And just in the last two slides, if I can compare this to the protein world, if in the magenta, you can see an RNA sequence, and in the yellow, these are stickers in the RNA that are allowing the RNAs to interact with the neighboring RNA. What we observed is that these stickers are embedded within the RNA through the RNA folding. And these are then leading to these transient and more dynamic interactions that we observe in granules. However, in repeat RNAs, these stickers are exposed, and there's a lot more of them as well, which allows for the formation of more persistent and strong interactions and the formation of disease. Of course, in vivo, these type of strong and persistent interactions can also be counteracted by the presence of RNA chaperones, including RNA helicases, which can convert this persistent and strong interaction into something much more dynamic, which is probably also something that is happening in RNA granules. And with that, I would just like to thank the lab, so Saran, Aisha, and Zichen, who did the work that I presented today, of course, our funding sources and our collaborators, the Hopkins RNA community. And again, I would like to thank you for inviting me here, Sonia, and everybody that was able to join for the seminar. Thank you.